Okay, guys, today we're going to work with mono hybrid crosses. I know you guys have watched the video that's kind of showed you how Mendel worked with these and some of the things that he did. And now we're going to work with monohybrid crosses, and we're going to do Punnett squares. And you guys are going to have a little practice sheet that you have to do. It's real short. It's real easy. You guys probably did this, I think, in 7th grade, but we're just going to go through it again and talk about a few things. The first thing, and I know this was mentioned in the video, the Ed puzzle you guys watched, is the difference in the terms heterozygous and homozygous. Homozygous, remember, means when homo is same, so that's when you have two of the same genes for a trait. So we're going to use mainly T's and we're going to use mainly R's. So the T's are going to be for tall, and the big T is tall. We usually give the symbol to the dominant trait. That's the one that's always going to show up if it's present. So two big T's would be homozygous dominant, and then for the little t, that's going to stand for short. And so homozygous recessive then would be two little t's. Heterozygous is when you have two different genes for a trait. So that would be if say we're talking about pea plants here, if they had a big T and a little t, they had a tall gene and a short gene. That's going to be heterozygous because there are two different genes for the trait, but that is still going to be a tall plant because tall is dominant, and so you only have to have one. On the other hand, for the plant to be short, you have to have two of the recessive traits, because if there's even one of the tall genes, then the plant is going to be tall. All right, so if we wanted to, and I'm just going to extend this page a little bit. If we wanted to do a cross, say, between, and this is what Mendel did, he took two pea plants that were homozygous for the trait. Another name that we sometimes use for homozygous is pure. And then a name that we sometimes use for heterozygous is hybrid. So if you ever see those are kind of more like older terms, but sometimes you'll still see those. So just in case you see those, you'll know what that means. And so what we would do to show this cross is we're going to do a little Punnett square there. And then we're going to fill in like what is on the top of the box and what's to the side. And we're going to do that for each box. Now you may say, well, why are we putting the capital T's first? You do not have to do that, but this is kind of like grammatically correct when you're doing genetics to always put the dominant trait first, because as you're reading left to right, it immediately shows you that that plant is going to be tall. Okay, so now that we have the square, we can actually find some information out about it. So we can find out the genotypic ratio. Remember, that's going to be the ratio of genes and genotypes within the offspring. So these kind of on the outside, these represent the parents, but on the inside, these represent the offspring that they would have. And something I think we get confused about is you think, well, they're only going to have 4. That's not the case. This could be 4,000. This is just what the ratio of those offspring would end up being as far as genotype and phenotype are concerned. So remember, genotype are the genes for the trait. So we're going to say how many of the offspring would be big T, be homozygous dominant. And as you can see, there's going to be none. So we're going to put a 0 in our ratio there. Now we're going to look up there and we're going to say how many are heterozygous. Well, we've got 4. So 4 are heterozygous. Then we're going to say little t, how many are going to be that homozygous recessive? There's none. So our genotypic ratio is 0, 4, 0. So now that we found the genotypic ratio, we also can find the phenotypic ratio. And that is a little bit different, because where we had 3 possibilities for genotype, we only have 2 for phenotype. Remember, phenotype is the physical appearance, what it looks like. And so in this case, we only have 2 choices, tall And they are all tall, so that's going to be 4. Or we could have short, and none are short, so that means our phenotypic ratio is 4:0. Now, something else that we'll sometimes ask, really probably even more than genotypic and phenotypic ratio, is sometimes you'll be asked percentages. Like, for instance, if I asked you what percent of the offspring were tall, that would of course be 100%. They're all tall. If I asked you what percent were homozygous recessive or had that little t genotype, well, none, so that will be 0%. None of them have that. Something else, we could ask for fractions. I'm going to try to keep it a little bit simpler since I'm not actually teaching you in person about this. So we're going to try to keep it more to the percentages, because I think that's easy for most students. You're either going to have 0, 25, 50, 75, or 100%. Okay, something else, we're going to do a real quick one here, is Mendel worked with peas that were round and peas that were wrinkled. So we're going to use capital R for round because that was the dominant trait. Little r for wrinkled because that's recessive. And then what happened, we're going to see what happened if he crossed 2 peas that were hybrid or heterozygous. So now this is going to look a whole lot different. Instead of everything looking the same, the parents once again are on the outside. Now we're filling in the boxes for the offspring. And so now when we look at our genotypic ratio, we have 1 that is big R, big R. We have 2 that are big R, little r. And we have 1 that is going to be little r, or homozygous recessive. So our genotypic ratio now is 1:1. Now phenotype, we still only have 2. We can have tall and we can have short. So if you look up here, you've got 1, 2, 3 that are tall, because remember, even though these have a tall and a short gene, a round and a wrinkled gene, and so I guess I need to change this. Sorry guys, this is round and this is wrinkled. Sorry. But it's still going to be the same math, if you will. So these, even though they have a round and a wrinkled, because round is dominant, it's going to kind of trump that, or it's going to hide that wrinkled gene. So we have 3, this one, this one, this one, that are round, and we have 1 that is wrinkled. Now, I may ask you, what percent are going to be heterozygous now? And if you look up here, you've got 50% that are going to be heterozygous, these 2. I may ask you, what percent are going to be wrinkled? And there's only this 1, so if each one's 25, then there's only going to be 25% that are going to be wrinkled. And guys, that I'm hoping will help you guys with your little worksheet. I'm going to give you a couple of days to work on this. It's super easy. There's only like 3 problems, and then I have you guys kind of distinguishing between hetero and homozygous. If you have any questions, just email me. I'll be glad to help you. Take care.
Will it take